I've been working on norovirus since 1998, and now my research is mainly focused on developing antivirals against norovirus infections. So we found by X-ray crystallography and some binding studies that the HMOs really mimic the HBGAs, so histoblood group antigens, structurally mimic. What they do is they bind to the pocket where the histoblood group antigens are, and the histoblood group antigens are a kind of cofactor for human norovirus. Some noroviruses require them for binding, and some don't. But the ones that require them for binding, what we found is that the HMOs bind to the identical pocket, and this likely would inhibit the virus from binding to the HBGAs. So you can find them in saliva, you can find them on the cell surface. So there's kind of two ways norovirus could interact with the HBGAs. They're first encountered in the mouth, the noroviruses with the HBGAs. Then they can attach probably to a cell surface where the HBGAs are expressed. So the HBGAs could be first encountered in saliva. So norovirus would first attach to the soluble HBGAs there, or they could be encountered on the cell surface. It's not completely known how or which ones they prefer to bind to and whether they actually are required for infection. There are a few studies that say that they are required, but the magical question is why some noroviruses require HBGAs and others don't require HBGAs for infection. We are dealing mostly with the human noroviruses, and we've done quite a library of different human norovirus strains, and we showed that most of them, I would say 70% of the genotypes bind the HBGAs. And if they bind the HBGAs, they're most likely to bind the HMOs as well. We recently found the G217s, which have been circulating in Asia quite a bit. They have bound the HBGAs, and they've also bound the HMOs. And more recently we showed that the HBGAs and HMOs can bind to quite distinct norovirus strains, so less than 50% sequence identity. So it's broadly reactive, the HMOs, to different genogroups and genotypes. So far we've just really looked at 2FL and 3FL. These ones are more predominant in the breast milk than the other ones. Something we'll do in the future is to look at other HMO types. But so far the 2FL, which as you know is quite a nice little prebiotic, is probably the best player and easier to produce than the other ones, mass produce. So really we want to concentrate our studies, future studies, on the 2FL, 3FL. Well, they closely mimic the HBGAs, especially the fucose, the way that's positioned on the 2FL and 3FL. And this is the main player in the interactions with the capsid, the norovirus capsid. But then you have the second and third saccharides, which are also important. They can be positioned and orientated differently and connected differently. But 2FL really mimics, in particular, a trisaccharide, for example. We would love to do some clinical trials with this 2FL. So that's in the pipeline, and hopefully that will go forward soon.